In this video, we will be talking about our August 2020 update and examining the global genomic epidemiology of COVID-19. For this report, we analyzed publicly shared COVID-19 genomes. Some of the regions we will be talking about are Asia, Oceania, Europe, South America, Africa, and North America. Since its emergence in late 2019, SARS-CoV-2 has diversified into several different co-circulating variants. We currently define 5 major clades. 19A and 19B emerged in Wuhan and dominated the early outbreak. 20A emerged out of 19A, dominated the European outbreak in March, and has since spread globally. 20B and 20C are large, genetically distinct subclades of 20A. We're now looking at the distribution of these clades across the world. You can see the countries in the Asia region have a higher proportion of 19A and 19B, which is seen in blues, as this variant dominated in the early outbreak. Europe and North America have a mixture of all clades, but are dominated by 20B and 20C. The D6146 substitution in the gene coding for the Spike S protein has been in the news recently. Evidence is building that the G variant, seen here in yellow, increases infectivity of SARS-CoV-2 in vitro and may have been evolutionarily selected for increased human-to-human transmission. However, an increase in infectivity may potentially come at a cost of making the virus more vulnerable to neutralizing antibodies. We can see that this variant was inferred to have appeared very shortly after initial zoonosis and subsequently spread around the world. In July, the 614G variant was present in the majority of circulating viruses worldwide. Since its initial appearance, the substitution has arisen multiple times and also reverted back to the 614D variant. There is no evidence that these other substitutions and reversions have resulted in continued transmission chains. As there are too many genomes to show in a single tree, we provide a regional analysis for each of the 6 regions shown here. In addition to our main global build, this allows us to focus on the diversity within each region while choosing appropriate out-of-region samples so we can maintain an overview of all the between-region transmission over time, as we can see on this slide. If we examine the situation in Asia from genomes collected before June 2020, we see signs of both extensive within Asia transmission as well as transmission to and from other regions of the world. Here, only countries in Asia have been colored, with the other regions represented by shades of gray. The color of each transmission line, the lines between the circles, represents the origin location, so all of the colored lines represent transmission originating from a country within Asia, in this example. This shows that many of the transmission involving Asian and non-Asian countries were importations into Asia. Especially pronounced in this view are those transmission from Europe to Asia. Though the lines seem to come from Germany, this point represents all of Europe. However, we must be cautious about how we interpret these inferred transmission, as sampling biases can play a large role, and we have many more samples from Europe than anywhere else. Looking at the genome sampled from June 1st, we see that the sampling is dominated by fewer countries. This limits the conclusions we can draw, but it appears that we may have fewer transmission between countries. This is also evident looking at the phylogeny, where we have large monophyletic groupings of genomes from Singapore, which is seen in yellow, and Bangladesh, which is light green. These data are consistent with the less recent international travel and stricter control measures. Shown here are only samples from Australia and New Zealand. You can see that the samples are spread throughout the tree, indicating that Oceania has been exposed to most of the observed genomic diversity of SARS-CoV-2. The majority of New Zealand samples, which is seen in blues, purples, and greens, come from a tight temporal band covering March and April, which is due to the successful control strategy employed by the New Zealand government. While the country is back to relative normality, the borders are still closed to all non-citizens to limit the chances for the virus to re-enter the country. Returning citizens must quarantine for 14 days before entering the country. Last week, the New Zealand government announced 4 new cases of community transmission which cannot be linked to arriving cases. Genetic sequencing may be able to help uncover how SARS-CoV-2 bypassed the strict controls. Australia and the state of Victoria, with the capital city Melbourne, shown here in orange, have been seeing a re-emergence of COVID-19 cases and have recently implemented further public health measures to try to curb this increase. These most recent genomes all appear to have the subclade of 20b. The recent dates and clusterings are signs of local outbreak. We can see similar clustering in sequences from New South Wales, where cases have also increased recently. New Zealand had reported over 100 days without community transmission before detecting cases in the community last week. As of August 14th, the cluster has now spread to around 30 known cases, primarily based in the largest city, Auckland. The source is not known yet. However, scientists have sequenced the isolates and reported that they fall into pangolin lineage B1.1.1. So while the genomes are yet to be released, they are known to fall into the region colored in blue here. This lineage originated in Europe, but has since been observed in multiple regions around the world. SARS-CoV-2 spread rapidly across Europe, likely primarily through direct transmission from Asia. By the end of February, even though there were just a few hundred cases officially reported in Europe, the virus had spread to at least 15 European countries. Given that sampling was less common in the early days of the pandemic, SARS-CoV-2 was almost certainly already circulating across most of Europe. Through March and April, much of Europe closed their borders, and many imposed differing types of lockdown where movement was restricted and businesses and schools closed. However, SARS-CoV-2 was already so mixed across Europe that different variants of the virus were circulating across multiple countries. Most countries had numerous distinct variants circulating before lockdown that were related to the virus circulating in other countries. This means the phylogenetic picture remains well mixed even after borders close. However, we can see some signs of the local transmission that we would expect. Finland and Sweden have a very distinct transmission cluster in green and orange, about 1/3 from the top, while Spain, dark blue, shows distinct local transmission at the bottom and the top of the tree. Iceland, in purple, and Switzerland in light blue also show clusters of local transmission. Examining only samples uploaded in the past week highlights two important points. Firstly, we can see a tendency towards groupings of tips into mini clusters. This indicates that within-country transmission is continuing to dominate, likely a product of the various regulations introduced throughout Europe. The virus continued to diversify genetically during the lockdown, but was most likely to be confined to one country. Some samples do not follow this trend of linking to other samples from the same country. For instance, in the middle of the tree, we can see some Swedish samples in green, nested within a larger Russian clade in red. Given the heavy subsampling, we need to remain cautious to not overinterpret putative between-country transmissions from this view. Secondly, we can see that the tips have a large difference in horizontal spacing. For example, the sample submitted in the past week represents a sample collection time window extending back to early March. The reasons for sequencing old genomes varies, but these samples help us fill in our understanding of viral evolution and geographic movement. South America's first SARS-CoV-2 sequences are from late February and early March, and are scattered across the tree, suggesting multiple introductions. As international travel decreased in March, we can see evidence of sustained local transmission in several countries. Many of Brazil's sequences, seen in light green, are part of two large clusters near the top of the tree, with some evidence that this variant also moved around the continent to Chile, Uruguay, and Argentina. We can also see distinct clusters of transmission involving Colombia in orange, Chile in turquoise, Uruguay in lighter blue, and Argentina in darker blue, scattered through the tree. Unfortunately, while SARS-CoV-2 continues spreading widely in South America, sequence generation has not kept pace. Though cases remain high across most of the continent, only 68 samples from 5 countries have been shared since May. Though our inferences are limited by the sparse sampling, in many cases, these more recent samples nest within the earlier diversity sampled in the same country. This suggests the other variants circulating now are descendants of those introduced early in the epidemic. Like South America, Africa had several introductions of SARS-CoV-2 to the continent. This is shown by the spread of African samples across the tree. Even samples from the same country include diverse variants. We now have sequences from countries across Africa throughout the epidemic. Though sequencing efforts seem to have declined slightly more recently, South Africa has contributed a large proportion of the sequencing. In the DRC, Senegal, and South Africa, we see clear signs of local transmission, shown as clusters of sequences in the tree. From Senegal and South Africa, we also have samples collected more recently. These samples generally fall within the older diversity of the country, as we would expect from the continued local transmission. Though we must be cautious with our conclusions, as they are greatly limited by highly biased sampling. The recent samples from Africa we have do not suggest continued importation of variants from elsewhere. This finding likely reflects the continued restrictions in movement globally. In mid-April, all U.S. states were on lockdown. States have since implemented heterogeneous reopening policies. In this tree, just like the trees for other regions, we see examples of local virus transmission as shown by similarly colored tips clustering together over time. However, on the tree, we also see within-country transmission across the US, as shown by mixing of colors at the tree tips. On the map, within-country transmission looks like the transmission lines extending between states. These observations are consistent with few domestic travel restrictions and states' reopening policies. There has been limited sequencing from Central America, especially over the past 2 months, which restricts the inferences we can draw about the epidemic. From the genomes that have been shared, we see geographic clustering of SARS-CoV-2 genomes. Toward the bottom of the tree in clade 19b, we see a cluster of transmission in Panama starting in mid-February. Several later sequences in Panama from June and July nest in with this cluster, suggesting there is continued local transmission of this virus genotype in the country. In Mexico, we also see clear geographic clustering of transmission through May. Without more recent sequencing, we cannot make inferences about the epidemic in later months. In the center of the tree, there is clusters of cases in Baja California in light blue. These cases were likely imported from California, USA in March. The green branches on the tree. This inferred importation is consistent with known travel links between the neighboring locations, although we should be cautious about interpreting inferred transmission locations given sparse sequencing. As the pandemic reaches its 8th month in global circulation, we can now see distinct changes over time in the picture painted by viral genetics, largely determined by changing behavior. Multiple early transmissions to the USA, Oceania, Europe, and across Asia were initially incredibly well mixed, as our global travel patterns distributed the virus incredibly effectively. Soon afterwards, diverse introductions to South America and Africa, often from Europe and North America, seeded local epidemics there. Borders are now reopening, and travel has resumed, though not nearly to pre-pandemic levels. This combined with the lag time from when samples are taken to when they are available publicly means we only see a few signs of between-country mixing. In general, recent samples tend to continue to cluster with past sequences from the same country, indicating continued transmission of previously circulating variants. Unfortunately, many countries have seen a rise in cases in the last few weeks, usually associated with reopening. As the Northern Hemisphere prepares to move into autumn and colder weather, further reopening and school resuming, we must remain vigilant about rising case counts. As winter progresses in the Southern Hemisphere, we see worrying signs that seasonality could indeed lead to more trouble containing transmission. Australia's initial success with the virus has been dampened by the recent outbreaks. Continuing to scale up tests, trace, and isolates, careful hand hygiene and conscientious mask wearing can help keep our societies as open as possible while we continue to battle SARS-CoV-2. We would like to acknowledge the amazing and timely work done by all scientists involved in this outbreak. Only through the rapid sharing of genomic data and metadata are analyses such as these possible. We encourage you to visit our website and click on the Explore the Data yourself, and scroll down for a full list of authors. The author of each individual sequence is available by selecting it on the tree.